Okay, so now we can start. We have Lenny also there. So Lenny is joined, so everyone is now inside. Okay, so good afternoon, everyone. Today we have Lily Dara with us. She's a physician scientist at the University of Southern California in LA. And I'm going to pass the word to Einar Björnsson, who worked with her, and he knows her much better than me. So you can introduce her if you want. Go ahead.
Yes, thank you very much, Gudvar. I had the pleasure to work with her on a meet the professor or meet the investigator breakfast at the AASLD meeting, and it was a very charming person and a very clever one. She has as our chairman told us, she's a physician scientist, and she has an animal model of different liver diseases. For example, immune checkpoint inhibitors induced liver injury. And just before I came to the meeting, I recruited a patient with ipilimumab and nivolumab induced liver into the ProEuro study, which is orchestrated from Malaga. And I told this lady that I was going to see a very exciting part of physiological mechanisms from Lily. So she's also a fantastic reviewer. So she's been reviewing papers for Journal of Hepatology, and I'm really looking forward to see how she develops in the future and to this talk. So thank you for joining us.
Thank you for the introduction. Thank you for the invitation. I am excited to be here and share my research, and none of this is published that I'm showing you. We're actually writing the paper right now, hopefully submitting it this month. So I'm going to share my screen. I'm going to move you to the left side so I can see your faces. I'm going to share my desktop here. And let's go to presenter view. Okay, do you guys see the presenter view?
Yeah.
Perfect. Okay. Thank you again for inviting me. I'm in Los Angeles, and I studied doing liver research a little late in my career, actually. I started during residency, mostly clinical stuff. I saw a few patients with drug-induced liver injury while I was rotating with Dr. Garcia Sao, Dr. Jim Boyer, and Joe Lim at Yale, and that kind of got me interested. And during fellowship, I met Neil Kaplowitz, and I had not done any bench research at that point, but Neil convinced me to go in the lab, and so for the past 13, 14 years, I've been a bench scientist. But my approach is very translational because early animal model, because I'm a physician. And I'm going to talk to you today about a little bit about overview of the immune checkpoint inhibitors and how they work, which I'm sure a lot of you are familiar with, and how we try to make this mouse model, and I'll show it to you, that gets a liver injury from immune checkpoint inhibitors. I'll give you a little bit of background because what I study is cell death mainly, and I'm trying to figure out how these cells are dying. And I'll finish up with some immunophenotyping that we've been doing, which Javier saw at AASLD. I presented some of that already this past AASLD. So basically, to start off, I want to talk about how these molecules work. As you can see here, you have T cells that are activated by antigen-presenting cells. Antigen presentation actually occurs through major histocompatibility complex 2 molecules, which are recognized by the T cell receptor here and result in T cell activation. The B7 ligand binding to CD28 on the surface of these T cells further modulates T cell activation signals. Now, CTLA4 here binds to B7 ligands in a competitive way to basically dampen this activation, right? As you can see here, it blocks. PD-1 and PD-L1 interactions actually diminish mature T cell activity after activation occurs. And basically, what immune checkpoint inhibitors do is that they block these inhibitory signals so that T cells can be activated against tumor cells. Because tumors are clever. They upregulate ligands to dampen the immune response. And so by inhibiting these, you actually get more T cell activation. And I know you're all aware of the liver as an immunotolerant organ and how it's a site of immune privilege. But just a brief overview of why we can model this in mice and why humans get liver toxicity. It's because these ligands, both PD-L1 and CTLA-4, are pretty active in maintaining the liver's homeostatic state of tolerance. So on the left-hand side, this is from a review we did a couple of years ago. I'm just going through this very briefly. On the left-hand side, you see that the constant exposure from gut LPS in the liver drives this state that is called endotoxin or LPS tolerance. This continuous low-grade exposure upregulates anti-inflammatory defenses in the liver and causes the secretion of, as you can see here, IL-10, IL-27, TGF-beta, increases FOXP3 T cells that are actually anti-inflammatory, and basically drives this immunotolerant phenotype. It also affects dendritic cells and Kupffer cells, and all of these cells kind of promote an immunotolerant environment. Non-parenchymal liver cells have been clearly shown to express PD-L1, ligand 1. Its expression in the hepatocytes is a little bit more controversial. There's studies that show actually hepatocytes do express it and some that show that they don't. So kind of the jury's out on that. But it actually is very clearly expressed on liver sinusoidal endothelial cells, Kupffer cells. So it plays a role in the liver. And on the right-hand side, another way that immune tolerance is maintained in the liver is by suppression of cytotoxic T cells. Basically, instead of fully stimulating T cells, what happens is that antigen presentation in the T cells is, in the liver is incomplete. And so both Kupffer cells and actually hepatocytes can do this too. They activate T cells into a little bit, and then they kind of force them to go through clonal exhaustion and anergy. So they actually, hepatocytes can act as APCs and activate these T cells to exhaustion. And this is a process that's characterized by upregulation of negative regulatory immune checkpoints, TIM3, PD-1, CTLA-4, and this ultimately leads to T cell death. So when you eliminate, these cytotoxic T cells, that is a good thing in terms of immune tolerance. So why did my lab start studying immune checkpoint inhibitors? For many years, I've studied acetaminophen, and I'm still interested in that. But what ended up happening is that as these drugs got approved, it became clear that they are very, very important in treating cancers. Survival from melanoma went from 6 months to 6 years now. So they're really important tools in the medical armamentarium. All oncologists use them. Many cancers are approved. They're approved for many cancers. But they do have drawbacks, which is the immune-related adverse events and the hepatotoxicity, which Ari and others coined immune-mediated liver injury from checkpoint inhibitors, or ILIC. I'll call it from now on because I think is the best description of the term. It has been a common occurrence and has resulted in patients who actually need the drug having to stop it. And elevations over 3 times the upper limit of normal, or grade 3 to 4, which are what we care about the most, often result in discontinuation of the drug. And when you have double checkpoint inhibitors, this can go up to 20%, the side effect. And these aren't going away. There's newer checkpoint inhibitors constantly being approved, targeting different pathways, and this is going to remain an issue. So understanding the underlying mechanisms is very important. This briefly, I just want to show you because I know probably like the DILI, you're also recruiting a lot of idiosyncratic cases. So there have been, and Jack Utrecht does this, there have been models of using immune checkpoint inhibitors to model idiosyncratic drug-induced liver injury. And that is not what I'm going to show you today, but I'm just going to briefly touch upon what Jack does, what he does is that he uses this CTLA-4 and PD-1 inhibition or blockade, or using knockout mice to sensitize a mouse to another drug. As you all know, when you get idiosyncratic drug-induced liver injury, many of these patients do have a specific HLA molecule or, MHCs associated with them. I don't know, B3501 for green tea, DRB11501 for augmentin. But there's many people out there in the world that have HLA DRB1 star 1501. They take augmentin and they don't get liver injury, right? And one of the hypotheses is that these people have a failure of immune tolerance. Most people would actually maybe get a little bit of injury, but their liver would adapt and dampen that immune response. And that's why they don't get DILI from augmentin. But those that do get DILI, they maybe have like this second hit, this problem with maintaining immune tolerance. And what Jack does is that he basically exploits this. And he had this really nice paper in 2015 that Neil and I wrote an editorial on, where he gave the mice that were PD-1, he gave them a CTLA-4 inhibitor, and he tried first with the drug amodiaquin. He's done this in the middle, as you can see, with isoniazid and nevirapine. He's done it with green tea extract. Basically, what he does is if you give the mouse just amodiaquin or isoniazid, they kind of get a little bit of injury and then they adapt, they get better. But if you give it to a PD-1 mouse that's also treated with a CTLA-4 inhibitor and continuously you expose them to this drug, eventually they get an injury from the drug. So they can't adapt to the continuous exposure to the drug. So this is a model where the checkpoint inhibitors are used in conjunction with a known idiosyncratic toxin. That is not what I'm going to show you. Others have tried to make this model, but usually they use either PD-1 knockout mice or wild type mice. There's a study that used B6 LPR mice. I'm just showing you this because we did a lot of research and we tried to figure out what dose people use inhibitors and how they've done this. And we settled on our own model, which I'm going to show you. Going forward, one other point I want to make in my background is that there was a really interesting paper in the Journal of Hepatology in 2021 by Lucia Posamai. The first author is Gud. And they looked at some human blood samples of patients that had LEC. And they actually found that there was a clear activation of macrophages and CD163 positive macrophages, which were homing to the liver. They had liver homing signals. They were CCR2 high and CCR7 low. It was a very interesting paper because it showed a role of monocytes in the liver injury. And that was something we were interested in studying. And I'll show you how that relates to our work in a bit. But I just want you to remember this because I'm going to show you mouse liver that looks very much like this. This is a CD68, which is staining for macrophages. This is CD3 for lymphocytes and a CD8 for cytotoxic T lymphocytes in particular. And a couple more slides on background before we go forward, just so you can follow my talk. Cells die in different ways, and I know that Javier studied this before, and he's an expert also in this field. Cells die by, especially in the liver, mainly apoptosis is the dominant form of cell death, and this is a complicated pathway. I don't need you guys to know everything about this. I want you just to know that apoptosis, no matter how it's activated, via which pathway it goes through, it's finally executed by cleavage of caspase 3, which is a protease that chops up stuff in the cell. So when you see cleaved caspase 3 activation, you know that the cell's mode of cell death is apoptosis. Necroptosis, which is a lot more controversial in the liver, depending on what model you look at, it is executed finally by this protein, MLKL, and phosphorylation of MLKL is necessary for the execution of apoptosis. There's other proteins involved in this pathway, RIPK1, RIPK3, but these proteins are multifunctional and they do other things and they're important for inflammation. So you really need to see phospho-MLKL activation for necroptosis. And for pyroptosis, which is a form of cell death mediated by the inflammasomes, you see gastromin D cleavage as the executioner of pyroptosis. You also see IL-1 beta cleavage, but this is what we look at to see this mode of cell death. And the NLRP3 inflammasome is actually the most studied inflammasome in the liver. It is a multi-protein complex. It's really important in regulating innate immune system and inflammatory signaling. It oligomerizes and activates caspase-1, which then cleaves IL-1 beta and activates IL-18. It can lead to pyroptosis, and it's very well studied in ASH and NASH, and it's been linked to liver fibrosis. So what we did in our model is that the collaboration with one of our rheumatologists who studies lupus, we got these mice that are haploinsufficient in CTLA-4, meaning they're missing one of the ligands. So CTLA-4 knockouts, they don't survive past a few weeks, but the haploinsufficient mice that are positive, negative, at baseline, they don't have a phenotype, but they are somewhat more prone to inflammation. And so we kind of took advantage of that, and we gave these mice, first we made a model with just the PD-L1 inhibitor, which I got from Ira Melman at Genentech, which is a mouse-specific PD-L1 inhibitor, and it's pretty potent. And then we added on the CTLA-4 inhibitor to kind of get a more robust phenotype. We also injected the mice that were controls with the matching IgG isotype. We tested the regimen at 1 week, 2 weeks, and 4 weeks. We did 2 injections, we did 5 injections, we did 7 injections. And we decided based on the injury we were getting that we were going to settle on the 2-week course. And here you can see that if we give the mice, IgG or isotype control, and we euthanize them, and we do 5 injections and we euthanize them at day 14, we can see an upregulation of this increase in this inflammatory infiltrates in the liver. As you can see, this focus here. These mice have higher ALTs, and we measured this, over surface area of the liver, and we quantified it, and they actually have a clear cut inflammation. What's interesting is that when we looked at their sera, they clearly had increased inflammatory cytokines, IL-1 beta, as you can see here, along with TNF, interferon gamma, and IL-17 was almost significant, also upregulated. So the first thing we were asking is, okay, we see this ALT, we see this injury, we see this inflammatory foci, what hepatocytes call, hepatopathologists call necroinflammation, conglomerating in the liver, and these areas, obviously the hepatocytes are missing because all these infiltrates are occurring. How are these cells dying? And so because apoptosis is the most common way hepatocytes die, that's the first thing we looked at. And as you can see here, when caspase-3 is cleaved, apoptosis is activated. On this Western blot, you look down here at 19 kilodaltons, you see that when you give immune checkpoint inhibitors, abbreviated ICI, you see cleavage of caspase-3. And this is significant. We've done this many times. We did immunohistochemistry on the far right here, and there's some hepatocytes that stain positive, and some of them are shrinking, dying, just like what you would expect for apoptosis. And this is a mouse that is given fast ligand as the positive control, just so you can see the hepatocytes, how they kind of shrink and die. So this matches. And then we try to do immunofluorescence to kind of figure out where these are. And I don't know if you can see this, maybe it's more obvious here. This is a binucleated cell that is dying. The other ones are kind of apoptotic bodies already dead. Maybe this one still has a little bit of its nucleus left as it's shrinking. But these really looked like hepatocytes. These don't look like immune cells. So we were pretty convinced that we're seeing hepatocyte cell death here. But we get into it a little bit further down as you'll see how we went about to show this. One of the things we looked at was necroptosis. I've been looking to find necroptosis in the liver, but I study acute models, and in acute models, it's been hard to find. I have all the tools, but unfortunately, I was unable to find any, as you can see down here, phospho-MLKL cleavage. So the mode of cell death did not seem to be necroptosis, although there was some RIP3 increase. But as I told you, there was a lot of immune cells infiltrating the liver, and the immune cells are very RIP3 positive. So I'm assuming this is a marker of the inflammation that was occurring because the end protein was not phosphorylated here. And we actually took some MLK-L knockout mice and we gave them the checkpoint inhibitors, and we did see liver inflammation in them. So we don't think that the mode of cell death was necroptosis. Since we were also interested in the inflammasome activation and pyroptosis, we looked for inflammasome activation. And interestingly, although we had clear evidence of apoptosis, we also saw the NLRP3 inflammasome was active. And as I told you, the NLRP3 inflammasome is activated when the innate immune system is activated. So that was interesting because we just talked about how the checkpoint inhibitors activate T cells, right? But it shows you also evidence from Posamai's group that they saw that in humans, there was some innate immune cells activated during LEC. So when we looked at these markers, we saw NLRP3s up, we saw caspase-3 was cleaved, we saw upregulation of IL-18. And so we couldn't quite see pyroptosis. This was not significant, but we saw some of the elements of the pathway are activated, especially the inflammasome when mice get immune checkpoint inhibitors. And when we looked at where this is occurring, I hope the resolution is high enough. Red stains for NLRP3. And here, if you look, you see the whole like sinusoid lining is positive for NLRP3. And that's probably Kupffer cells, which are macrophages, and they all express NLRP3. But also you see these, big blobbed hepatocytes that are also expressing NLRP3. They're random, but some of them are associated with these areas of inflammation. So that was interesting because we know that the inflammasome marker NLRP3 is very much expressed in macrophages, but we also saw it in hepatocytes, as you can see here. So we were trying to figure out what's going on here. What is the role of caspase-3? And we had a tool that was really interesting, is that we have a collaboration with Ionis Pharmaceuticals, and they make antisense oligonucleotides, which are used to knock down genes. They have actually FDA approval for a drug for hypercholesterolemia. But I will tell you this, they gave us two types of antisense. One that knocks down caspase-3 in the whole liver. So it knocks down caspase-3 in hepatocytes, in the LSECs, the non-parenchymal cells, any immune cell that's in the liver. And they gave us another type of antisense that targets specifically hepatocytes. And I'll talk about that after this. So here you're looking at the antisense that basically decreased caspase-3 in the entire liver cell population. I lost my mouse. Okay, here we go. So we designed this, we tested this out a couple of times, and then we came to the conclusion that we're going to knock down caspase-3 for 2 doses, and then we're going to give the immune checkpoint inhibitor, and then we're going to alternate it with the antisense. We did, dose curve experiments. We settled on a dose that we had previously determined. And we saw that, yes, if we give caspase-3, we can knock down the antisense, we can knock down caspase-3 in the liver. So you'd think that these liver cells couldn't die anymore because you block caspase-3. Well, we were wrong. They still died when you give them immune checkpoint inhibitors. And actually, it was kind of worse. The injury in a lot of the mice, as you can see, it's scattered, biological replicates. If anybody who works with mice knows that you can never get a completely clean result. There's a kind of like a confidence interval, right? A spread. But when you do, 12, 15 mice, you see actually clearly that the caspase-3 antisense seems to make everything worse. And definitely it's not protecting. So that was curious. And this could partially be explained by the fact that we are not, by giving caspase-3 antisense, we're immortalizing all the immune cells and kind of, so they're secreting more inflammatory cytokines and making the injury worse. But anytime you block caspase-3, you have to look for necroptosis. That's a given. Anytime you block apoptosis, I didn't have time in my 40 minutes to go through the whole background of cell death, but that's when you see necroptosis, is when you block apoptosis. But when we looked for it, we actually didn't see phospho-MLK activation. So again, we couldn't see any evidence of necroptosis. That was really curious. When we went on to do this other type of antisense, which is called GalNAc, conjugated antisense, just a brief background on this. These are these antisenses, abbreviated ASO. They promote degradation of complementary RNA via an RNase H mechanism. So this company has a drug called Kinmaro approved for homozygous familial hypercholesterolemia, and they were trying to test it for that purpose, but now they kind of like in collaboration could make it for any target. They use this asialoglycoprotein receptor, which is a C-type lectin that's expressed on hepatocytes very, very abundantly. And when they conjugate the antisense to this, it has a lot of affinity for the N-acetylgalactosamine or GalNAc moiety. And so when you conjugate an antisense to this GalNAc, it is picked up by the hepatocytes and not by the non-parenchymal cell. So it improves the potency of the antisense. You can use a lot lower dose and you can target hepatocytes. So we did dose curves on mice. You can see why this is not published yet. This is years and years of in vivo work. We did dose curves on mice, and we found a really low dose, 1 mg per kg, that was effective. And as you can see here, I'm just going to show you, these are the non-parenchymal cells, the liver white cells, and they're pretty much equivalent, whether you give a scrambled antisense or this caspase-3 targeted antisense. But then when you look to the right, if you give this GalNAc caspase-3 antisense, when you compare the hepatocytes to the hepatocytes that got the control, there's clearly knockdown, right? This is not a complete knockout, but it's enough to decrease the amount of caspase-3 inside the hepatocyte. And so what we saw is that when we gave this, we actually saw a protection overall, and by ALT in particular, in the liver. They still do get inflammation, but it's a lot better than it is in the control immune checkpoint inhibitor. So when you target hepatocyte-specific caspase-3, you don't get that problem that we saw previously where the injury gets worse. So it seems like targeting caspase-3 in the non-parenchymal cells is not a good idea. And because we didn't see necroptosis activated, we went on to look for what's going on with pyroptosis. And we actually have been seeing that with the global caspase-3 liver knockdown, the unconjugated version that made everything worse, we actually seem to see, look, if you can look here, cleaved caspase 11, oops. You can see Gas-German D and terminal. So markers of pyroptosis, and as you can see here, NLRP3 is highly upregulated here in the caspase-3. It goes up with both with all the ICI treated mice, but it's significantly up if you knock down caspase-3 in the entire liver. So it seems like when we try to block apoptosis, and also affect the immune cells by blocking apoptosis, we're actually hyperactivating, immortalizing them and hyperactivating the inflammasome and the pyroptotic signal. So this is what we have going on so far, but we also wanted to figure out what types of infiltrates are in these mouse livers. And so we took areas where we took some liver slices and we did immunohistochemistry on them. And we saw that, yes, there are CD4 positive cells, there are CD8 positive cells, as you expect in these immune infiltrates because you're giving checkpoint inhibitors. But there's also a good amount of F480, which is a marker for macrophages, activated as well. We wanted to do some cutting edge research, and this is what we've been doing for the past few months. We were trying to figure out how we can do immune phenotyping on tissue using single cell technologies, but also trying to understand relationships between cells. As you all know, these are all like newer platforms. They're very expensive. They're hard to do. And so after doing some research, we settled on doing image mass cytometry. Image mass cytometry is basically a cytometry by time of flight on tissue. It uses stable, isotopically distinct metal tags that can be easily distinguished in cells by atomic mass cytometry. And we're using these metal probes as opposed to fluorochromes because they are less, I want to say bleedy, there's less overlap with them. These aren't non-biological and non-radioactive metals. you can multiplex, I don't know, 40 different tags at the same time. And so it's really interesting. But one of the things that's really crazy is that, the mouse antibodies are not fully developed. So we had to do a lot of trial and error. These panels are very well developed for humans. But here just showing that, fluorescent labels, anybody who's done IF knows there's a lot of overlap and you get a lot of, background and false positive signals. But with IMC, because you're distinguishing these tags by their metal mass and mass spec, basically, they're very distinct, and there's less. It's not impossible, but there's a lot less overlay. And so there's no need for compensation of these overlapping signals. And each vertical bar here is a stable isotope that can be measured. So how does this work? You stain the tissue with the specific antibodies that are tagged to these metals. And then you have this, you insert it into this machine. It has a motorized stage, which then there's a chamber that fills with helium gas. There's a UV laser that blasts the tissue. And these basically antigen complexes fly off like plumes from the tissue, and they go through this chamber by argon pushes them through to a machine, which then what happens is they pass through this, they get ionized, and they fly by time of flight. They separate based on how heavy they are, and it gets vaporized. And eventually they hit the detector at different, as you can see here, they based on their mass and how heavy they are, they hit this detector. So the detector can understand that, for example, CD4 was attached to this metal that is lighter, and CD8 was attached to this metal that is heavier. And because the machine had the coordinates of that UV laser that was precise at 1 micrometers, it can actually tell you where that antigen flew off the tissue. So it's very fascinating how this occurs. So what we did is this is like the panel we built. We tested it. These are the metals. They're tagged to different antigens. This is how you conjugate it to the metal. What we then did is we made a tissue microarray like they do for tumors, but these are just liver cells. There's no tumors. And then we kind of did an H&E, and we found the areas where because this was random punches, where the inflammatory foci kind of were occurring. We ended up actually capturing a lot of this. We ended up we also captured some normal tissue as a control. And here you can see what this looks like. So for example, this is an H&E. You see in the inflammatory foci here. Hopefully the resolution is good enough on your screen. But if you can simultaneously, for example, I've turned on here 1, 2, 3, 4, 5, 6 channels, and then we can get an overlay of where they lie. You can see CD4 here. This corresponds to this H&E, right? These are consecutive slices. They're not the exact same slice, but they're literally right after this slice. Here you can see CD8 and beta-catenin, which is staining hepatocytes, and CD11b, which is staining innate immune cells. And here, when you overlay them, you can see that these are all co-occurring in the same focus. So we really were interested in cleaved caspase 3, and when we turned on cleaved caspase 3, as you can see here, cleaved caspase 3, which we think is the marker for the dying hepatocytes, is also occurring around this area. And so we see both innate immune cells and, for example, here you're looking at T helper cells accumulating in this area where there's hepatocyte dropout. Now, you can mathematically quantify these by obtaining single cell data. You put on a single cell mask. This is not me. This is we collaborate with a data scientist. You define the nucleus, and you actually, as you can see, isolate each cell. And when you do this, you can actually, because you have 1 micrometer precision, you can and the cells are what, like, a hepatocyte is big, right? Like 50 micrometers, and maybe the the immune cells are smaller, I don't know, 5-10 micrometers, you can actually, this cell, how many of these antigen antibody complexes are inside each cell. And so it gives you a pretty precise understanding of how the cells are located within a tissue, which cells are interacting with the dying hepatocytes. And obviously you can't conclude causation, but you can conclude associations, right? And so the first thing you do is you get all the single cell data, which is enormous. And the data scientist gave us these basically phenotyping bar graphs and heat maps. And so in order to, the first thing we had to do was basically do phenotyping and clustering and to identify. So he showed us, he said, what do you think these cells are? What do you think this cluster is? And so we went through the markers and we said, okay, this is a hepatocyte. This is a hepatocyte. This here, for example, is a non-parenchymal cell. It looks like it's a FOXP3 positive. So these are Tregs, these are CD4 cells. So we identified these clusters of cells, and we went on to take out the hepatocytes and do subphenotyping of the non-parenchymal cells. And this is a UMAP showing the non-parenchymal cells and their distribution, showing where each cell lies. And if you turn on different markers, this is a better way of depicting it. For example, if you look here, this is a B cell marker, B220. This island here is showing some markers of B cells. The CD3 cells are showing up here. So these are all lymphocytes, and also here, right? So this population is lymphocytes and this population is lymphocytes, but the CD4 ones, right, are showing up more here, this cluster. So you can go on and you can figure out where each phenotype is occurring. And then when we compared the mice that got immune checkpoint inhibitors to the mice that were controls, and obviously these mice also have these markers, they still have immune cells in the liver circulating in the sinusoids or just residing in the parenchyma, you can see that they clearly delineate and distinguish themselves from the control mice. And there's more CD8 cells, CD4 cells, and innate cells, neutrophils, plasma cells, so obviously more inflammatory foci, and a lot more proliferating cells active in the immune checkpoint inhibitor mice. Now, this is what's holding up the paper. We're actually doing analysis this month, trying to understand the relationships between these cells and the relationships between these cells and cleaved caspase-3 positive cells and NLRP3 positive cells. Are these the same cells? Are these neighbors? And are they interacting? And there's mathematical ways of doing a nearest neighbor analysis to understand cell-cell interactions and to kind of give an idea as to which cells are in the neighborhood and vicinity of the dying cells and how these cells are interacting with one another. And the trick is to kind of like show this in a nice kind of figure so that it's understandable to everybody. And so we're working on that. We have some of that data, and hopefully we're excited to submit this paper next month because we really need to. It's been a long time. And I just want to, oh my God, the names, when I changed it, didn't show up. I have to probably come out of this just to show you. I don't know why this is white. I guess I changed the font and they didn't, my postdocs. Here we go. So this is Layla, who is and now a resident. She did a lot of the in vivo work. Jacob, who is a fourth-year medical student going into internal medicine residency. They both want to be gastroenterologists, hepatologists, and physician scientists. This is Helia, who is in my lab now. I want to thank, all our collaborators, my mentor, Neil Kaplowitz, my whole mentoring committee here, and then people who have helped a lot with this, with the IMC particularly, and then my funding, including the Dylan, who's funding me to actually do this study in human liver samples who got immune checkpoint inhibitor toxicity. And that's it. I hope I'm not over time. Oh, it looks like we have some time for questions and answers.
Yeah, thank you very much, Lily. It was impressive, very impressive data. Einar, you want to start or
No. You take the question.
Okay. I have a lot of questions, but because I think this is really novel. I mean, just to model, no, ICI, immune checkpoint inhibitor, drug-induced liver injury is quite difficult, as you show. There has been some work, no, with PD-L1 knockout mice. Then you showed yourself the CTLA-4 heterozygote mice. You mentioned also LPR mice. Have you tried these ones as a model for lupus, as you know?
Yeah, no, I haven't. But if that paper, the Adams paper, I can send it to you. They tried V6 LPR mice and they got a good phenotype.
Yeah, this is what I'm saying, because in my hands, you get, I mean, the problem of these mice is that you have CD4, CD8 cells in all the lymphoid organs that they accumulate, no? So you don't know whether the damage comes from the mice itself or from the ICI, you know, from the immune check, from the drug, you know. What I really like from your presentation was apart from this scheme with the pyroptosis versus apoptosis and necroptosis, is that when you use when you model the DILI in your mice, you have an increase in caspase-3, but also an increase in inflammasome, no, like such as NLRP3. Is that right? But you focus then on caspase-3. Why you didn't focus on NLRP3? So what I mean is that you then go ahead and try first to knock it down, then to use GalNAc. But why don't you say, okay, and now we have also, because you had all the inflammasome markers upregulated, no? What you didn't know is whether it was hepatocytes or immune cells.
Yes.
Would have not been easier to do, for example, flox mice for NRP3 hepatocyte specific knockout mice, or CLEF4, for example, for Kupffer cells, and induce the damage there?
Yeah, so that's actually in the works. We're trying to use the issue was I wanted to go for something that was kind of like the end of the pathway to kind of get a clean result. The problem is if you knock out an NLRP3 high up, you really don't know exactly are you just preventing the entire inflammasome, or is this pyroptosis, you know, because there's so many things that could happen downstream. But it is in the works. I actually, you know, I'm a hepatocyte biologist, so I was focused on targeting NLRP3 in hepatocytes, so using antisense next, hopefully, but trying to get this version of the paper out, because if I go after that, that's another year of work. And you know what I mean? But and it's like I'm at that point in my career, you know, this is the Young Investigator series, that I just need to publish this so I can apply for my R01, and I can't apply until until I publish this. So it is actually something that we've discussed actually with my mentoring committee, that yes, I need to do that, but first get this paper out. I like the idea of doing it in a CLEF4 floxed way. Maybe looking at I'm not sure exactly, because I see it in the hepatocytes clearly, but I also see it in the Kupffer cells, and most people report NLRP3 in Kupffer cells, you know.
Absolutely, I agree.
But I don't know why I'm seeing it upregulated in hepatocytes. You know, I mean, on immunofluorescence, for what that's worth, but I do see it in hepatocytes too. But it's scattered. It's not as much as the Kupffer cell. Okay.
Just another question. And I pass. When you block a caspase-3, either with the GalNAc or, well, you did it at the end of the GalNAc, but did you check for other caspase effectors, or how if they go up, or like, for example, caspase-7 or caspase-9, if there is any, like for example for Jung, no, when Jung1 or Jung2, if you just block Jung1, normally Jung2 goes up, and vice versa.
So maybe that's what explains the fact that caspase-1 and 11 are up. But we looked at what I thought. We looked at 1, 11, and 8. I don't know. 8 didn't change. It was just a really hard antibody to use. I haven't looked at 7 and 9. but that's a good thought. I can look at them.
Okay, no, it's just
yeah.
because I understand that to catch these proteins by Western blot is really a hell, as usual.
Also, like, it's a snapshot in time, right, Javier? That's the
Yes.
problem with this. Because like you do this experiment for weeks, and then it's when you kill the mouse, how many cells do you see that are that moment dying and are positive for cleaved caspase 3? And that's what I'm trying to like tell the data scientists I'm working with. He's like, so these cleaved caspase 3 cells, do you know what other markers they're co-expressed with? I'm like, they're dying. Everything is chopped up, you know, like I don't know what I don't know how you're going to co-express it with something. It's very hard to study, and especially because it's not like fast ligand where you have like massive apoptosis everywhere. This is scattered, right? Like these ALTs are like 80, 90, 100 max, you know, like 120. They're not going to be like massive, you know, they're not like acetaminophen where the ALT is like 5,000, you know?
Exactly. But I think in spite of that, I think it's quite novel. It's quite novel work. So no, very nice work. Any other questions? Please go ahead.
I could just pick your brain, Lily. Excellent presentation. Really impressive. Very nice. But just to try to pick your brain, going back to trying to connect the clinical world to your work and comments you made in the beginning. So when you look at the DILI world, we have maybe 3% of the patients, maybe 6% sometimes, that actually get liver injury, and maybe 90%, more than 90%, do not get liver injury. And can you speculate? Immune tolerance is a theory that is very strong, but many of those patients do not have even a minimal ALT elevation. It's not like they develop an injury and then develop immune tolerance. So the question is, do you have any speculation based on your work and what you're involved in on why that is?
Yeah, I actually do. Actually, the mice, if you look, we had to do like 15, 18 mice because not every single mouse gets the injury, right? So it's kind of like it's a staggered, you know, if you look at the dots, like some of the mice are much lower, some of the mice are much higher. But on average, when you have a lot of mice in each group, you can see that there's a trend that when you give immune checkpoint inhibitors, those mice do get the injury. So two ideas, and I've been thinking about this a lot. One is either the immune checkpoint inhibitors in some people are bringing down the immune tolerance threshold enough that you get a response to something else, you know, like the dacarbazine plus an immune checkpoint inhibitor. A lot of these patients are on other medications. It could also be, so either we're sensitizing to something else, and these patients maybe have a sensitivity to drug X or Y that's causing the reaction, but we're just decreasing the threshold, or something in the environment, I don't know. Or two, I wonder if there is a dose threshold, you know, a threshold for immune tolerance, and the people's thresholds are different. To kind of, you're just not blocking it enough in some people versus other people. And then, you know, the contribution of how these checkpoint inhibitors actually affect innate immunity is not studied enough. You know, we've been putting on blinders and saying, oh, we're activating T cells. We're doing a lot. We're activating FOXP3 T cells. We're depleting, FOXP3 T cells. We are affecting other cells, you know, these are important for antigen presentation, and maybe there's something to be said about people's innate immune system. I know that, you know, Bob Fontana and the Dylan are also looking at, you know, HLA associations for these patients, but I'm kind of starting to get involved with that too. I don't know if that's going to be enough, because this is not a classic, you know, this is not an idiosyncratic DILI kind of phenotype. But it's complicated. Obviously, these are all speculation, but it could be that it's like a combination of having the right HLA plus, you know, the threshold, or you're sensitizing to something else on top of that. But it's, what's interesting though, the reason I say the threshold is because why is it that when we give 2, we get so much worse injury? And I see it in the mice too, right? Why is it when you block both, when you increase the potency, you get more injury? So it must have to do with some threshold.
Thank you. Excellent talk.
Thank you, Lily. Very inspiring and also provocative thoughts and idea. The first thing I want to tell you is that for me, it has been, now that we are handling a paper, the denomination, how you called. You said for you, the best way is DILI, which sounds, well, it's easy to pronounce, no? We have used Chile. Someone suggested that the nomenclature that likes he most was CPILLI, which is very difficult to. It should be something like simply or something like that. So I think it would be good to come together and try to have one unique terminology.
Like standardize it. Yeah, I think Ari can speak to this. They tried doing this. They wrote a paper, a consensus paper. And Ari, didn't you guys discuss how you're going to name it?
Oh, for months. We've had so many discussions, and yeah, we ended up preferring to leave Chile for food substances.
Yeah,
so, but yeah, since it's such a specific reaction, we thought that checkpoint inhibitors should be in the name until we figure out if there's a more general phenomenon. So that's why we thought checkpoint inhibitors should be in the name of the phenomenon.
Yeah. Okay.
Yeah, I just went by that also because they published it and they specifically kind of thought about it and kind of consensus wise decided on that name. So I'm using the same name in my, clinical papers and and my and also in the mouse model.
You're using DILI. No?
Yeah. One of my residents is doing a retrospective review on our checkpoint inhibitor patients with HCC who got DILI, and we're going to submit to an abstract to the liver meeting, and we're using DILI. We're just going to try to stick with one name.
One name, yes. And the other thing I wanted to ask you, because this is it has been always intriguing, no? That once you get an injury, the patient gets re-exposed again to the drug, and then you may come up with a liver injury, but sometimes or most of the times they seem to adapt, no, to the injury. They do not develop liver injury.
So, I mean, it depends on the grade. For a lot of them, they just come off of it altogether. But, some of these patients, more and more, I'm seeing the oncologists, they have discussions with us and they're like, well, we need to start it, it's going to die of melanoma. And actually, usually they tweak it a little bit. Maybe they change to another agent or something. And I will say there's data on this, I think Naga, I don't know who published this. I want to remember, I don't remember right now. There's an American journal.
Miller and Naga, Miller.
Yeah, yes. And they had, I know it's amazing memory. They had 30% reoccurrence out of the ones that were reintroduced. So 70% do fine.
Yes. Well, I had this patient today who had been on nivolumab for 1 year without any elevation, and then ipilimumab was introduced, and after 3 courses, he developed a very high elevation rate.
Yeah, so actually, when I looked at our data, there was a clinical trial for ipi-nivo in HCC, and those had the highest immune checkpoint inhibitors in toxicity, and they actually had more immune checkpoint inhibitor toxicity compared to the melanoma patients. The HCC, obviously, they have underlying liver disease, so that's kind of like the abstract we're going to submit. So kind of because also we don't know, in patients with underlying liver disease, right? I'm sure it's a different kind of immune microenvironment, and so we kind of really need to look at that too, compared to other things. But yeah, the double, I agree with you, Einar, the double is definitely more toxic.
Yes, for sure.
Maria, how many immune checkpoint inhibitors do you guys have in your cohort?
Do you remember, Aina? Cannot
it a
tell
lot?
you. I think we did not look at that.
Yes, we
Group.
have in the I think it was around 30 cases or something in the
Yes, it's true. It's true. Immune
publication.
checkpoint inhibitors. I can send you the paper, Lily, that was published from the Pro-Euro DILI. Yes. So you can find out the precise figures.
Perfect. I think I may have seen it actually. But do send it to me. I would love to look at it again.
Perfect.
Excellent. Any other question? Because we are already it's 9, no, in the morning in LA.
Yes, it's 9:05. I don't know. I'm happy to stay if there's more questions. If not, thank you for inviting me.
Yes, yes, I
Well,
think there
Lily,
are no more
can I
Yeah,
take
Einar.
2 minutes of the time after the other people have left, because there was a case, Investigator hydroxycut I had.
Oh,
other
really?
day. And I'm going to discuss it with you, but we can spare that discussion for the others. If you can
Okay,
stay
yeah,
on the line for 1 or 2
I can
minutes.
stay. Yeah, sure, I can.
Okay.
Ismael is telling me that there were 33 cases.
33 cases. Okay.
The memory wasn't that bad. 30.
Yes.
33.
Anyhow,
Okay.
we'll send you the manuscript. Yes.
Perfect.
Okay, Lily. So thank you very much for giving us this very impressive talk, and we are very happy to have you here today.
Thank you,
So,
Javier. I'll see you in Boston.
I will see you in Boston. Yes, for sure. Okay.
Thank you.
Bye.
Okay.
Bye.
Thank
Bye,
you.
everybody.
Thank you, Lily.
Thank you for joining.